So this is going to be another two-part talk. Um, I'm in the SANS lab, and we are a membrane lab, which I think is different from what a lot of people here are doing. And so my talk is going to be giving the context of how we like to think about membranes and lipids and why mycoplasma and SYN3 are such good model organisms for the work we're trying to do. And then Natasha is going to get more specifically into the research she's doing. And so the guiding question that I hope to set up, and then she'll start to answer a little bit, is why does life need so many lipids? So now I'm going to hop in. We know that cell membranes are essential to life. They're conserved across all life as we know it, and they have highly conserved properties. Cell membranes have to be semi-permeable. They have to allow in some molecules and allow out some molecules, but they have to be selective in which they allow in and out. They have to be robust. Cell membranes are the interface between the internals of the cell and an outside environment which can be harsh or toxic. Cell membranes have to be responsive. The outside environment is always changing, and the membrane has to respond to those changes to keep the internals of the cell working as they should. In order for the cell membrane to be responsive, it has to be dynamic. Cell membranes don't have a steady state. They're always adjusting their lipid composition so that they can respond to changes in the external environment. And finally, something we take for granted is cell membranes are lipid-based. All the membranes that we know are primarily composed of lipids, and there's a reason why life chose this path, or why this path was the most favorable evolutionary path for life. Even though we know all these properties of cell membranes, we still don't understand them the way we would like to. And specifically, we don't understand the link between lipid membrane composition and cell fitness and cell functionality. And really briefly, for anyone who hasn't worked with lipids in a while, I'm going to give an overview. Most of the lipids in cell membranes, specifically the mycoplasma membranes, are phospholipids. They have a polar head group, non-polar fatty acid chains. This property means they like to self-assemble into bilayers so that their hydrophilic head groups face a liquid and their hydrophobic tails can face inwards. These acyl chains can be saturated or unsaturated, meaning that the carbon chain has a double bond. And one of the reasons that life really likes lipids is because there's a tremendous diversity in lipid classes and lipid species. So lipids can have different head groups, and different head groups have different sizes and also different charges. They can also have different saturations, saturated, unsaturated, or polyunsaturated chains which change the size of the lipid in the membrane. They can also have varying numbers of chains. Lysolipids only have one chain, while some species of lipid, like cardiolipin, have 4 distinct chains. And finally, the number of carbons in the acyl chains can be different, and this will adjust the thickness of the membrane. And this means that life has access to a tremendous toolkit of lipids with which to build its membrane. Cell membranes are fluid mosaic. Their structural component is primarily lipids, and there are sterols, which is a very hydrophobic type of lipid interspersed in the membrane, which make it robust to environmental changes. In mycoplasma and SYN3, these sterols are cholesterol. And finally, you have both peripheral and integral membrane proteins, and these proteins need a tightly defined microenvironment to function properly, and that environment has to be provided for them by the lipids. And the lipids determine the membrane properties which allow the proteins to function. Like I said, the length of carbons in the acyl chain determines membrane thickness. Head group can determine both the local and the global charge on a membrane. And then the specific properties of the lipids, as well as the temperature, determines the membrane fluidity, or how tightly or loosely lipids are packed together and packed around the proteins. And in response to changing environmental conditions, membranes will remodel their composition. They'll switch the lipids in the membrane in order to maintain a tight microenvironment for the proteins. And to do this, membranes like having lots of lipids. So most eukaryotic and prokaryotic cells have hundreds of different lipid species. And life has invested a lot evolutionarily in having highly complex membranes. We know this, but the complexity of the membrane makes it difficult to understand. It's hard to identify levels of regulation or changes in the composition of one specific lipid class or lipid species when you're just dealing with such a large amount of complexity. And that's why we in the lab are hoping to simplify the membrane so that we can better understand it. And mycoplasma and SYN3 are excellent model systems for simplifying the membrane. They have small genomes, which means they have fewer levels of regulation than some more complex organisms. They have a single plasma membrane, they don't have a cell wall, and they don't have organelles. So it's easy to access the membrane. And you also know if you're talking about lipids in mycoplasma or SYN3, you're talking about the plasma membrane. They're a mammalian parasite, which means that they need to take up nutrients from the host cell, and one of these nutrients is lipids. Unlike most eukaryotic and prokaryotic cells, mycoplasma and SYN3 cannot synthesize most of their lipid species. They cannot make acyl chains, and so they rely on either their host cells or the diet we feed them in order to get the lipids that form their membrane. As I said, they have a simpler membrane than most eukaryotic and prokaryotic cells, but this membrane is still more complex than we would like. And one of the things that you're going to hear from Natasha is how she can make this membrane even simpler by very carefully picking which lipids we feed the mycoplasma. However, even when we define the diet we feed the mycoplasma still has the ability to remodel the lipids in its membrane. And I said that mycoplasma and SYN3 cannot synthesize their own acyl chains, which is true. What they can do is use phospholipases in order to break down lipids that they're fed and then use the components of those lipids to synthesize a couple species of lipids. So they do have some remodeling capability. I'm going to keep this as an overview, but in general, there are 4 families of phospholipase active in mycoplasma and SYN3. You have phospholipase A1, which cleaves an acyl chain in the R1 position, phospholipase A2, which cleaves an acyl chain in the R2 position, phospholipase C, which cleaves the head group before the phosphate. However, this seems to be involved in small molecule signaling rather than remodeling lipids for membrane composition. And finally, phospholipase D, which cleaves the head group after the phosphate. So even with these very simple diets, mycoplasma and SYN3 are able to dynamically remodel their lipidome. And that brings me to my research, which is still ongoing, which is, can we push the system even simpler? Can we remove some of the remodeling activity in mycoplasma and SYN3? And the way that I hope to do that is through diether lipids. So on the left, you see the lipids that mycoplasma would normally have access to, and they have ester bonds, which can be attacked by phospholipase A1 and A2. Instead, I work with synthetic ether lipids that have this ether bond. So while phospholipase A1 and A2 are active when, or at least are functional when you have linkages, when you work with diether lipids, you can knock out virtually all phospholipase activity in mycoplasma, and not knock out in the genetic sense, but you feed them a substrate that the enzyme can't attack and can't break down. And so initial results show that it seems like I've pushed my system to the simplest it will go. I've pushed it to a system with under 10 lipids in its membrane. And that's when we start to see the system break down. And when we're working with a system so simple that the fitness of the organism suffers, we can start to figure out the answer to the question, why does life need so many lipids? So I'm just going to leave you with the 2 points that are driving my research. First, pushing living membranes to their simplest viable point, to the point where they start to break down, and using that to identify a link between membrane composition and the fitness of the cell. Thank you for listening, and now I'm going to turn it over to Natasha, who will speak to this in more detail.
Questions for Isaac?
I would propose that we keep questions until after both talks because we're speaking to similar things.
Natasha, you're up.
Do you see my presentation?
You're all set.
Nice. Okay. Okay. Hello, everyone. My name is Nataliya. I'm also a student in James Lab, and I will pick up on a general introduction that Isaac just gave you and try to show some of the results that we have obtained in our lab so far. So just a quick recap, one of the fundamental questions in membrane biology is why do membranes need so many lipids? We know that we can construct a robust synthetic membrane using just one lipid species, while living membranes on average have over 100. So mycoplasma and SYN3 are very useful organisms for us to try to create a minimal membrane model system which will have tunable lipidome size and composition. So in order to do that, we started with looking at the lipid composition of animal serum FBS that is historically used to feed different mycoplasma strains. And from this analysis, we found out that the main lipid classes in FBS are cholesterol, phosphatidylcholine, PC, sphingomyelin, and a couple of lysophospholipids. So when comparing the lipid composition of animal serum with the mycoplasma membrane lipidomes when they are grown in this animal serum, we identified that mycoplasma in general picks up all the main lipid classes from the serum, with cholesterol taking up to 2/3 of its membrane. They also like PC, sphingomyelin, and at the same time, they have two different lipid classes, phosphatidylglycerol PG and cardiolipin, which are not present in the serum. And this suggests that they synthesize these lipid classes internally by taking up free fatty acids available in the serum. So with this initial layout, we can switch to trying to create defined lipid diets by taking cholesterol, which we know is absolutely necessary to support mycoplasma growth, also adding PC and sphingomyelin that they like. And on the other hand, we can also try supporting their internal lipid synthesis by adding fatty acids so they can make their own PG and cardiolipin. Then the question is, which fatty acids do we need to choose so that they can induce their own lipid production? From literature, we know that the most common fatty acids used by mycoplasma are palmitic and alleic acids, one saturated and shorter and one more unsaturated and longer. And they show to make mycoplasma able to produce their own PG and cardiolipin. So these lipids make our general lipidomic pool that we use defined lipid diet in mycoplasmas. So we started with the most complicated defined lipid diet where we basically feed them all of the lipids that I just mentioned. So cholesterol by default, PC, sphingomyelin, and two fatty acids. So that means two fatty acids, POPC, sphingomyelin diet. We also experimented by removing either of these external phospholipids in the diet, POPC, for instance, or sphingomyelin. And finally, we came to trying the minimal possible diet in this setup when we have just cholesterol and two fatty acids. So now, when we have all of these diets, we fed them to mycoplasma, and the first question that we asked was how changes in this lipid diet that we give them changes their overall lipidome size. And by lipidome size, I mean the number of structurally distinct lipid species in the lipidome. So if we start with FBS, cells grown on FBS as a reference point, we see that their average lipidome size is about 100 lipid species. when we switch them to the most complex defined lipid diet with fatty acids, POPC, and sphingomyelin, we drastically decrease the lipidome size to under 20 lipid species. It's almost a 5-fold change. So sequential removal of either phospholipids in the diet gives us the opportunity to reduce the lipidome size just a little bit more. When we came to the most minimal diet with just fatty acids and cholesterol, we realized that the minimal cell doesn't grow on this diet. So just 2 fatty acids are not enough to support the minimal cell growth. Mycoplasma mycoides grows pretty well on this diet and produces the minimal possible lipidome of just 11 species. So far, I've talked about adding or removing the external phospholipid classes in the lipidome, but there is a different approach we can take to modify lipid composition of mycoplasma membranes. And instead of adding phospholipid classes, we can support their internal lipid synthesis with more fatty acids. So if we start with the 2 fatty acids minimal diet, we see that they have one saturated and one unsaturated fatty acid. And what we can do is to provide them with a complementary set of fatty acids, with the longer one being now saturated and the shorter unsaturated. And this is a new 4 fatty acid diet, which has a distinct feature of decoupling the acyl chain saturation and acyl chain length for mycoplasma, thus giving them more degrees of freedom in constructing their lipidome. So when we fed this diet to the mycoplasma and minimal cell, which shown here in green, we figure out that both can grow well on this diet. So 4 fatty acids is enough to support the minimal cell growth. And this diet also produces the largest lipidome in mycoplasma among all the defined lipid diets. So this plot essentially shows you the main output of my entire PhD project, which was to construct a minimal membrane model system with tunable lipidome properties using mycoides and Syn3B. And now using this system, we can really dive in and study how cell membrane works. And to support this claim, I'll show you 2 things where we link the lipidome size to the membrane properties adaptation in this minimal membrane model system. So to start with, we do know that in response to temperature, all living membranes remodel their lipidomes to maintain biophysical properties and their bioactivity. And by lipidomic remodeling, in this case, I mean the sum of the total change in all lipid species abundances within the lipidome with temperature. So if we plot the lipidome size on x-axis this time and the overall lipidomic remodeling on the y-axis for both mycoides and Syn3B that were grown on all the defined lipid diets in FBS, we suddenly see a negative correlation between the lipidome size and the overall lipidomic remodeling, which suggests that membranes with larger lipidomes are more robust and do not need a lot of changes to maintain bioactivity of their membranes, as this result is conserved in the minimal cell. This indeed suggests that this is a fundamental properties of the living membranes. And the second point that I wanted to show you is based on the notion that all of the living membranes need to adjust their biophysical properties at lower temperatures to maintain their bioactivity. And one this fundamental biophysical properties is membrane viscosity. So what we did to see if the viscosity adaptation is different for mycoplasma membranes with different lipidome sizes, we grew cells on all of the diets and measured their membrane viscosities at 37 and 30 degrees with the example viscosity values you can see here of cells grown in FBS, and took the difference in viscosity estimates across 2 growth temperatures to obtain this result. So what you essentially need to know to understand this plot is that in case of a perfect adaptation of membrane viscosity to temperature, cells should exhibit no change in the absolute viscosity value as the temperature goes down. So the smaller change means better adaptation to temperature. And you can see on this plot that both mycoides and the minimal cell, the larger lipidomes on 4 fatty acid diet, 2 fatty acid PCSM diet, and FBS produce much smaller change in the membrane viscosity with temperature. And this once again reiterates the importance in larger lipidome size and membranes in their ability to adapt to temperature. So with this, there are two main points that I want you to take away from my talk. One being that in our lab so far, we have managed to create a minimal membrane model system with tunable lipidome. And the second point is that by changing the lipidome size, we figure out that larger membranes have greater robustness in response to environmental perturbations, and larger lipidome size also helps to maintain membrane biophysical properties. With this, thank you very much, and I welcome your questions.
Very nice. Do you think your work addresses issues that may look back to the primordial evolution of bacterial membranes? I know mycoplasmas are not at all primordial species, but we may be recapitulating something here. Either of you.
I think yes and no. I think the yes comes from the fact that it seemed logically it would make sense that primordial membranes would be simpler membranes than we have today. Especially if you can imagine that a lot of the machinery that cells now have to remodel lipids would not have existed in a primordial system. So I think the idea of studying how a very simple membrane behaves both speaks to primordial life, and I also think it's essential to think about when we want to move forward with building synthetic life. Because the more you can reduce the number of components you need to build a living system, the easier it will be to both build that system and to computationally model that system. The no is that the composition of the mycoplasma membrane is very similar to that of mammalian cells. So cholesterol, for example, did not exist in primordial times. Other parts of our lab work with hopanoids, which are essentially analogs of cholesterol, but have a much simpler synthesis process. And they might be better analogs for primordial membranes. And actually, there's a member of the lab trying to grow mycoplasma on hopanoids. So that might bridge that divide.
Certainly, as you think about Andre Saki and Chris Dyke's work, that if you want to try to make a synthetic cell using a synthetic membrane from chemicals off the shelf, that your work maybe would be a guide as to how to make that, because it seems like if you made your synthetic vesicles that didn't recapitulate much of what is in a modern, in your, at least your simplest membrane, that you might be killing yourself, even though you create a functional TXTL system and a genome inside a lipid vesicle. It might not work because you've got the wrong lipid composition.
Absolutely
Yeah.
agree.
One thing I would add to that is that, we know a lot about how mixtures of 1, 2, or 3 lipids behave, how lipid structure influences property, but we know almost nothing about how mixtures of 10 or more lipids behave. And it's from these experiments that Natasha and Isaac are doing are really emphasizing how it's important that we begin to understand the emergent properties of complex lipidomes. And what that means for how membranes work, rather than just simple in vitro systems.
Could a protein or a lipid interactome or a protein lipid interactome be made?
Yeah, actually, one of the things Isaac's going to work on with Andre Nodler here in Dresden is lipid protein cross-linking pull-down experiments. We have a method for that set up. So that's something, and that's really important in identifying the sensors and regulatory components.
Just have quick, I'm just wondering, is there any effort going on to start adding other pathways, like, to have the cholesterol? Again, I will bring the E. coli plasma again, which doesn't need that, right? So the media is super cheap. They can live without this. And again, this would make it even easier for everyone to grow. So is there any effort going on to add some of those pathways to not have the requirement of cholesterol added to the media?
Well, like Isaac mentioned, I mean, the hopanoids are one possibility. This is a sterile analog that requires a much simpler pathway that we could potentially put into the mycoplasma because it's so simple. And so, it does seem like they can probably grow in those alternate sterile analogs. So that would be one way. I was wondering, the mesoplasma, I read somewhere it didn't require sterols. I wonder but I don't know how well that's been verified, and there were a few older papers I think that suggested that.
Yeah, I don't know if Sebastian is here still, but yeah, I've been growing some of it in the lab, and I think we were adding a little bit horse serum, but yeah, E. coli plasma, we don't add anything, and they are they're growing perfectly fine, and
Yeah.
with Mesoplasma, I think so.
Yeah, I mean, the reason we didn't really work with the coloplasma is just because they have so much more complicated lipidomes. They have glycolipids and carotenoids, or they can take up carotenoids, right? It seems like a bit more of a complex problem, although they are simpler in the sense that they don't require so many things from the media. But yeah, getting them unaddicted from sterols would be a nice step.
I can remember papers by, I think, Shlomo Rotem years ago that they were able to wean wild-type Mycoplasma mycoides or Mycoplasma capricolum. They're, I think, for our purposes, basically the same. They were able to wean it from cholesterol, although the cells hated it.
It's also, so the paper you're referring to, there's one where they did successfully wean it from cholesterol to hopanoids. When you try to grow it without any sterol, they could get it to low cholesterol. They could never fully wean it off of cholesterol. It needed a certain amount of sterol in the membrane to stay alive.
It's true that you can reduce the cholesterol amounts to like tenfold lower values than we usually feed them to make them happy. But also then there is a trade-off between the cholesterol level and how well they actually grow and how easy it is to do anything with them when they're so much deprived of the sterol.
Can I ask? Excuse me, can I ask? This is Shige Kakizawa from Japan, and thank you for your excellent talk. And maybe I missed your part of your talk, but how about the growth rate of the SYN3 without FBS, adding the cholesterol and so on?
Yeah, so I didn't focus on that today. So what I can tell you is that on defined diets, they actually do not grow worse than on FBS for SYN3. I think you see a bigger difference in growth rate for mycoides, but SYN3 grows equally bad on most of the lipid diets that we feed them, including FBS. And surprisingly enough, at lower temperatures, like 30 degrees, defined diets actually support SYN3 growth even better than FBS does. So
about the price of the SP4, it is very expensive because of the serum is too expensive. So based on your result, maybe the media price will be drastically decreased. Is it right?
It's definitely lower I cannot tell you the precise difference in costs. I think James would be a better person to answer that question. But I do know that if you switch them to defined diets, it's definitely cheaper than feeding them on cereal.
And actually,
Thank you very
the
much.
lipids is the cheapest part. It's the we use BSA as a substitute for serum to solubilize the lipids, and the BSA can be quite expensive because we usually buy it delipidated. So. But there are potentially other ways to get around that we're exploring, but it's a bit messy at the moment.
Thank you very much.